Thanks so much for having me join my hostess spotlight video for everyone. Great. Just quickly, my conflicts, I'm on scientific advisory board for a therapeutics company. I'm not going to talk about therapeutics, and the company does not work in the autism space. What I do want to do today in the next 20 minutes or so is really just drive home a couple of points and observations. The first is that over the last decade, large genomic data sets have resulted in reliable and systematic gene discovery in autism and other neuropsychiatric disorders. I focus on autism today. And so I'm going to spend the first few minutes just talking about where this has gotten us and what we now understand about the genetics of ASD. And then I'm going to move to talk about how we might be able to use these molecular clues now that are quite substantial, combine those with large omics data sets in order to begin to understand what the underlying mechanisms might be of disease, and particularly to see whether or not we can use those large data sets to begin to distinguish kind of the pleiotropic biology of individual genes from a core pathophysiological mechanism, or one of a core pathophysiological mechanisms for autism. So because I'm concerned about the time and have a tendency to run long, the most important slide, these are the folks that have done all the heavy lifting here. You're going to see their names repeatedly, particularly Stephen Sanders, who's associate professor at UCSF now, and Jeremy Wilsey. They trained in my lab and are leading wonderful independent research labs now, but continue to collaborate on some of this work. And then I also want to highlight Veronica and Belinda, who co-authored the piece in neuropsychopharmacology, which I hope you have a chance to take a look at. I want to start just by saying that the key observation about a decade ago in autism genetics that has really led to the lion's share of progress during that time was the understanding that de novo, or new, spontaneous mutations contribute to ASD. And what I have here on the left side, I'm not sure whether you're looking on the left side, is a pedigree that's pointing out the dynamics of a de novo mutation, which is that the mutation is not present in either of the parents. It typically happens in the germline, for germline de novo mutation, in sperm or egg just prior to conception, and then is present in a single offspring, but is typically not, or is not present generally in their siblings. And these mutations have some very interesting properties. One of them is that if you're interested in mutations that have large effect, de novo mutations, as I said, happen just at fertilization. And as a consequence, natural selection has a very limited time to operate on these. So you can have very large effect mutations leading to structural brain abnormalities, significant challenges with regard to IQ, et cetera. But as long as the pregnancy comes to term and you have the ability to identify these new mutations, you can see in that first generation the connection between a mutation and an outcome. And these outcomes can be sufficiently severe that if you waited over several generations, those mutations would generally be purified out of the population through purifying natural selection. So there's some very strong interest on the part of several groups at the turn of the millennium in the contribution potentially de novo mutation. And the first technology that allowed us to look for those genome-wide were microarrays that allowed us to look for submicroscopic changes in chromosomal structure, to do this across the genome and to use it in that first generation to be able to see whether or not there was a connection between de novo mutation and affected status, in this case, autism. And by about 2007, 2008, Mike Wigler and Jonathan Sabat showed that in families in which there was only a single affected individual with autism, that there appeared to be, even in small cohorts of just a couple hundred people, a difference, an overrepresentation of de novo heterozygote losses or gain in chromosomal structure in affected versus unaffected. By 2011, several groups, ours included and Mike Wigler's, published on sort of the first large-scale analyses of these, looking across the entire genome. We're able to replicate those findings and then to use this overrepresentation of these heterozygotes deletions and duplications in the genome to begin to identify regions of the genome that were carrying risk for autism. Fortunately, technology was bounding ahead, and it was only a year later that multiple groups were able to use a similar approach, looking at an affected sibling, either against unrelated controls or against a sibling control, to look for these de novo mutations using sequencing, where we could sequence essentially every gene in the genome, called whole exome sequencing. And what we found, again, was the same thing that we found for these copy number variations, that de novo mutations, and particularly those that hit a gene and were likely to be damaging to the gene, were overrepresented, in our case, in probands versus siblings. It was also the case in probands versus unaffected controls. And again, the initial samples were relatively small. A couple years later, by 2014, multiple groups were looking at several thousand families, each with 3 or 4 individuals, so nearly 10,000 individuals, to find this overrepresentation of mutations that are heterozygous. They're on one of 2 copies of the gene, that are damaging to the gene. They're de novo, present in every cell in the body in the affected offspring, and not present in the rest of the family. Now, multiple groups were able to use this with the development of appropriate statistical tools to be able to differentiate those observations of these new mutations and see whether or not they were associated with the phenotype. By 2015, our lab had consolidated multiple consortia to pull together kind of the current state of the field with regard both to the individual genes that were being hit by these gene disrupting mutations and copy number variations. And then just recently, all the same groups now in an even larger consortium, down with about 21,000 people, or about 7,000 families, were able to identify now more than 100 genes that have strong statistical evidence as being associated with autism spectrum disorders. And I just want to point out that here in the blue, the genes about which we have the most confidence, so the strongest statistical evidence here with an FDR of less than 0.01, collectively to have a mean effect size of about a 20-fold increase in risk. So we were using de novo mutations to look for these rare events that carry large effects. And it turned out that they were there, they were present, they were contributing to autism spectrum disorders. So these are, again, coding mutations, heterozygous on one of 2 copies of the gene. They typically are leading to the gene, one of the 2 copies, having a loss of function and conferring very large effects. Now, at the same time, there were a number of groups that were using what is more typical of complex psychiatric genomics and what's happened over the last decade, which is to look through genome-wide association studies for common variations in the genome that carry individually small effects. And here in autism, it took actually quite a large group of patients, quite a large cohort, to be able just recently to get over the finish line and identify the first 5 loci. These are non-coding changes that are associated with risk. And now we're talking on the neighborhood of individually, maybe a 5% increase in risk for any individual loci versus, as I just described, for the top de novo mutations, 20-fold or greater increase in risk. And I'm pointing this out, sorry, this actually should say 5 loci, not 10, just caught that typo. And it's both, this is kind of where we are. And with regard to common variation, and I want to make a couple comments about sort of what that gestalt is versus our understanding of something like schizophrenia. So in schizophrenia, it's almost been the inverse picture in terms of looking at common and rare variation. So the lion's share of progress in schizophrenia has been looking for common variances. This is a seminal paper published in 2014 that found 108 loci from about 40,000 cases and controls. So that's about twice the sample size I just showed you for autism, where we were able to identify 5. Part of what this is telling you, if you think back to that last slide, is that any common change in the genome that carries a substantial effect on an early onset disorder like autism, one that's often associated with intellectual disability as well. That again, that natural selection works, and they are really driving down the frequency and presence of those. So the very small effect sizes are the reason that it's taking such large samples in autism to begin to identify the first handful of contributing loci. Although we do know that common variants are carrying overall in the population the lion's share of risk for autism. They just, as I said, have very, very small effects. Now in schizophrenia, inversely, so this is not even published yet, but it's from the Broad group, just put up on medRxiv. And what this is showing is recent gene discovery in schizophrenia using the same methods we used in autism starting 7-8 years ago, and now finally getting to the first 10 large effect de novo or loss of function mutations in schizophrenia. And here I want to point out a couple things about the slide. So this is showing, these are effect sizes. So again, very large effects. Although for schizophrenia, we had to get to 24,000 cases to get to the first 10 genes, and in autism, again, sort of the inverse story. So a couple things that I want to point out here about the genomic architecture of these two disorders, because I think they begin to give some insight into how sort of these large-scale gene discovery efforts might begin to open the door to understanding some important aspects of biology. So the first is, these are all the copy number variations that I mentioned in the first several slides. And these are showing the effect sizes in schizophrenia. And one of the things that has been so kind of vexing for the field is that while the slide I showed you that gave a list in autism clearly showed large effect mutations in autism, this is exactly the same set that I showed you on the second slide. So essentially every copy number variation that we identified in autism in 2011, essentially almost simultaneously were found to also carry risk, not only for schizophrenia, but in some individuals for intellectual disability without autism, autism without intellectual disability, bipolar disorder, ADHD. So there was a group of mutations early on in this discovery process that really called into question whether there would be any specificity at the molecular level at all for these kind of severe neurodevelopmental disorders. Now, what's happened more recently is that this set of 10 schizophrenia genes, the top ones that have been identified now very recently, there's zero overlap with 102 loci or 102 genes that I showed you previously for autism. And again, suggesting that while there certainly is some overlap here, and it's going to be very interesting to see where those overlaps are, it also seems as though we're beginning to see potentially some molecular specificity around different neurodevelopmental disorders, which I think will help us begin to think about how we might be able to leverage the specific molecular clues to find out pathophysiological mechanisms for different diagnoses. Because of the time, I'm going to skip over the whole genome sequencing. An interesting technology. The bottom line from that slide is we're probably about halfway there in terms of the number of samples that we need to get to a statistically significant result that will survive corrections. So I'm going to stick with what we know about autism genomics right now and talk about how we begin to think about moving from genes to pathology. So almost immediately, the molecular clues that I just showed you began to suggest, even just with a prima facie look at them, that they were not all over the place in terms of function. So two large areas of annotated function jumped out almost immediately. On the one hand, a lot of these encoded for proteins in the synapse, and a separate group encoded for proteins involved in the regulation of gene expression, often through chromatin modification. What I want to do, though, is just spend a couple minutes talking about some of the challenges of trying to understand pathology from kind of this level of understanding. I think what you can see is that these are really great diagrams, but they're essentially pointing to kind of all of biology. And I think one of the challenges that I want to highlight here is that we may have had an early kind of expectation that we could find a gene, understand what protein it encoded for, and then potentially what cell type was involved, and kind of move along a traditional sort of translational paradigm to go from gene to therapeutics. But the reality is that biology, unfortunately, is not that simple. And for each of our autism genes, they generally encode multiple proteins. Those can be expressed at different times in different cells, contributing to multiple circuits. And of course, for neurodevelopmental disorders, this is happening across developmental time, so that you get tremendous dimensionality here to anytime you move beyond the gene. There may be multiple protein isoforms that are determined by when you look and where you're looking in the brain. So this begins to suggest that following the path from gene to behavior, let alone to therapeutics, can be quite challenging. And then on top of that, if we're doing that and potentially, say, in a mouse model, by doing kind of the traditional, we find a gene, we knock it out, and we see what happens in the mouse, that we know that there's significant differences between mouse brain and human brain. So we may be more likely to have high levels of conservation here, kind of the closest to the gene, and more complexity as we move a bit farther away and towards complex behaviors. So what I want to do is just take you through an example of how we were thinking about using large-scale genomic data sets to try to see whether we can gain some additional traction on this problem. And here it's a pretty simple idea, which is that if you have the opportunity now to begin to really understand what the kind of molecular landscape is of human brain development, you could begin to ask the question, not what any individual gene is doing, but begin to take multiple genes, because we have a large set and a growing set of them, and begin to ask whether or not using these omics data sets, whether or not we can begin to identify points of potential convergence on the path from gene to behavior. So a fairly simple notion that these will overlap, not just in terms of what they do, in terms of, you know, being at the synapse or being involved potentially in transcription or translation, but potentially where and when they might be doing them. So I'm going to give you one example of this and then finish. This is from a collaboration with Anand Seston, who's been a prime driver in developing pictures of the transcriptional landscape, the gene expression landscape of developing human brain, using something called developing something called the BrainSpan Atlas. So here, what we were able to do is to take clusters of human brain region gene expression across windows of time. This is about 4 weeks of gestation. These are human postmortem samples, all the way to 80 years of age. And we took our highest confidence autism genes and we asked sort of what were they doing in terms of their interaction with other genes, creating networks called co-expression networks relying on this BrainSpan data, seeding them with autism genes and looking to see whether there were spatial and temporal kind of coherent networks of functioning. And then we asked, among those networks, if we took additional autism genes that were not in this original analysis and looked to see whether any of these windows of time and any of these brain regions had a statistically increased rate of genetic risk related to autism, where might that tell us that along this transcriptional landscape of developing human brain that risk might be coalescing? And in fact, we were able to find, somewhat surprisingly, I think, you know, we had an expectation. This is a heat map, and this is the minus log p-value. We thought this might light up the entire grid here. In fact, both we and Dan Geschwin's lab, who was kind of doing this at exactly the same time using very similar approaches, we actually landed in the same issue of Cell with this analysis. Both found that prefrontal cortex and glutamatergic neurons in mid-fetal cortex were the relevant spot where we were seeing at least the beginning of this coalescence. You can see here that there are potentially other areas of coalescence, and I'll talk about that as I give you my last slide. So here's another analysis, but unfortunately I didn't anticipate how long it would take me to get through. So this will have to wait. Essentially, the bottom line is we've now used 102 genes and found very similar answers with some additional specification of where this point of convergence is. So the bottom line is, my last slide, that we now have used large-scale genomics to identify multiple genes. And now with developing databases at the protein level, protein complex level, beginning to understand the cellular architecture with single cell omics technologies, we're starting to fill in a developmental profile along this continuum. And it allows us to take these individual genes and to look for coalescence. So these are systems approaches to large-scale omics data sets. And they, as I said, allow us to begin to think about, can we begin to understand not just kind of what genes do that are contributing to ASD or now schizophrenia or other disorders, but when and where they're doing them as an entree to begin to really understand what kind of pathology we might be able to target in the coming generation of therapeutics development. So I'm going to end there.